Hello, my name is Cymone Houston. I'm a civil engineering major here at Lamar University. My mentor is Dr. Thinesh, and my CERT project is the biological treatment of produced water. Produced water from oil and gas extraction contains harmful contaminants that are released into the environment. Treating and recycling this byproduct is costly, and storing it in underground wells is disastrous. Therefore, this research will develop a biological system using one strand of algae called Galderia sapphira to remove and recover the contaminants in the produced water. The specific objectives of this project is the characterization of produced water, the evaluation of optimal dilution in large-scale reactors, and also seen in Figure A, and the evaluation of nutrient and algal biomass production potential using produced water in lab-scale reactors, 1 liter, and you'll see that in Figure C. So why Galderia sapphira? The growth conditions of Galderia sapphira has high temperatures of 20 to 58 degrees Celsius and up to 68 degrees Celsius. You can see in the photo on the top right. It has a low pH of 0.2 to 4 pH on the pH scale, and it tolerates high salt and metal concentrations. This algal strand is also known to outcompete most conventional wastewater pathogens, and it's also generally transformable to deal with various chemicals in emerging conditions. In this slide, we will discuss the produced water characterization. In Table 1, you can see the specific characteristics of produced water. The pH of produced water is generally measured in the values of 6.71, and also the total suspended solids and the total dissolved solids that are in produced water, and also the high electrical conductivity of the produced water, with also an emphasis on the last part on the table, which is the high production of the ammonium nitrogen. In the chart to your left, you will see how Galderia sapphira was able to treat the produced water. This chart shows the asteroid weight of the algae, which is just the biomass of the algae, to how the produced water performed within its 7 days of running this test. You can see the high concentrations of produced water did not do so well. However, the 20% in the blue, the 10% in the orange happened to be one of the highest performing results we got out of the produced water being treated with Galderia sapphira. And you can see on your right the ammonia nitrogen removal, with day 0 being the red that you see, and the day 7 being the green. As you can see in the 5%, 10%, 20%, and 50%, almost all of the ammonium nitrogen was completely removed from the produced water. Now, if you move down to the phosphate phosphorus chart, which is right below the ammonia nitrogen chart, you will see that the day 0 in the red and the day 7 in the green happen to be the lowest in the 20% produced water sample. So when tests were run in our 1 liter reactors, the experimental setup of this test was that we had 1 liter tubular bubbling reactors. They had a 12 to 12 light cycle. They had carbon dioxide levels between 2 and 3%. And the experimental conditions of this is that we had 5 tubes of with the controlled media, and we had 5 tubes containing 20% produced water, which is based off the last slide that I said 20% produced water happened to be the highest results that we got of. So in continuation of the results that you see, we will use 20% produced water. This experiment was ran for 7 days, and the temperature of this experiment was kept at 42 degrees Celsius. The results from this 7-day experiment concluded that 20% produced water happened to show the best growth, and that the final algal biomass density of this produced water growth, of the removal of the contaminants within the produced water, showed that the control media only had 5.38 grams per liter of algal biomass density, and the 20% produced water had a 3.20 grams per liter biomass density, basically showing that the produced water was able to completely um, that the algae was able to completely grow within this produced water and thrive within this 20% produced water than the control media. So the continuation of these results from the 1-liter reactors is that the nutrient removal for the ammonia nitrogen, which you will see the graph in the top right, shows that the control media was that the ammonium nitrogen removal within the control media was removed 41%, but within the produced water it was removed about 97%, and you can see that in the bar graph to the top right, that the produced water on day 0 completely or almost completely removed the ammonia nitrogen. And you can see with the phosphate removal, the control media removed the phosphate about 15%, and the 20% produced water, the removal was about 33. So about 2 times removal, or the produced water was able to remove about 2 times as much as the control media. The future directions of this project would be to continue the experiments in 10-liter reactors to evaluate the nutrient removal and the biomass growth within these pilot scale reactors, and also to leverage the data obtained from these pilot reactors to optimize the system's performance in real-life applications. I would like to thank the Office of Undergrad Research and the Department of Civil and Environmental Engineering for funding this research project. I would also like to thank my research team, Melissa and Ari, and a special thanks to Dr. Baram for giving me the opportunity to be able to continue in the research. And also, I would like to thank my mentor, Dr. Thinesh, for helping me through the process.